Hello, everyone, and thank you for joining us today. We're very excited for this week's NeuroTools Webinar series. Today we have Dr. Paul Pavlidis with us today, and without further ado, I think I'd like to push the microphone over to Paul and let him get started.
Thanks, Devin, and welcome everybody. So I'm going to talk about some work in my lab on the title of Presentation of Cell Type Resolved Gene Expression and Applications to Neuroscience with a database and set of tools we developed that we call NeuroExpresso. And I'm a professor at the University of British Columbia in Vancouver, Canada. And since I'm not going to do video, there's a picture of me so you can have some idea. I can't see the people who are on here, so I always find these webinars to be a little bit of a weird experience because I feel like I'm just talking to myself to some extent. So I did tell Devin that if there are questions in the middle, to feel free to bring them up in the course rather than waiting for the very end. So I'm really excited to be able to talk to you about this work. It's still quite new, and I thought I'd start by giving a little bit of a background about my lab and sort of where we're coming from. And so my background is in neuroscience, originally training in neurogenetics and molecular biology, and I've been doing computational research purely for almost 20 years now. And in my lab, we're focused on the applications of computational methods to understanding questions about the brain, and we're very definitely focused on things that are. So hopefully you can see my pointer here. So that on going from on the gene side is where a lot of our efforts are focused in genes and genetics and genomics. So especially gene expression analysis. So I'll be focusing on that today. But of course, you want to connect these sorts of questions. Data types to questions at other levels of organization of the nervous system. So cellular phenotypes like connectivity or electrophysiology, and ultimately we want to connect this to understanding how the brain works in health and disease. And so at my lab, we do a lot of work on developing methods, databases, and tools. So that includes developing software. We do a lot of data curation. We apply a lot of statistics and machine learning methods, and we also apply the tools and methods that we develop to analyses of data sets that either are generated through collaborations that we have or from publicly available data sets. And I'll talk a little bit about some of the reanalysis work that we have done. So I'm only going to talk about one of our tools today, NeuroExpresso, but I just wanted to bring to the group's attention that there are a bunch of different things that we've produced over the years and which are still under active development. You can access those through the lab website. So the only one I'm going to be talking about is down here at the bottom, NeuroExpresso, which doesn't have a cute little logo yet. But other ones that we developed are GEMMA, which is a database of gene expression profiling data for meta-analysis, and ErmineJ, which is a software tool for doing gene enrichment analysis. So just to give a couple of examples. So we'll be talking about NeuroExpresso, and just to maybe connect things together to the first slide where I talked about integrating things across levels, is that one reason we collect all these different types of data and try to integrate them is so that we can answer questions that come up a lot in neuroscience research. So if you have a gene, you want to know where is it expressed in the brain. We want to know things like what sort of electrophysiological features it might be involved in, either directly or indirectly. We want to know about cell types and their relationships to diseases and genetics, how genetics influences phenotypes as well. So this is just one sort of slice of kind of questions that we're interested in addressing through the tools we develop and in our application areas. So the work I'm going to talk about today is it's been in development in the lab for a couple of years, but and it's still not fully out there for the public. So under in peer reviewed form. So for people who want to know more about the details, there's a preprint, and that's for the paper that's under peer review. We're hoping that this is going to be accepted, any day now. And this is also a good time for me to mention my my collaborators and coworkers. So a lot of the work I'm going to be describing was done by Ogun, who's a PhD student in my lab, and Leela, who is a postdoc with a lot of input from Sridhar Tripathi, another postdoc. And we've also been collaborating with Etienne Sabil's group at the University of Toronto with his postdoc Brad, who has done some of the validation work with us. And this is also a little bit of a preview for an SFN short course that I'll be participating in the day before the main conference starts. So this is on November 10th. And so maybe if there may be people who can maybe still sign up for this, but we'll be giving a session on that's going to be similar to what I'll be talking about today, but there's also going to be a hands-on component that people can participate in if they want to see more about how to actually apply the tools themselves rather than just working it out on their own. Okay, so just now to get to the meat of it, so this is an overview of what I'll be talking about. So what our motivation is here, so challenges in interpreting brain transcriptomes, how cell type resolved expression can help, and then I'll talk about NeuroExpresso and how we built it, and then I'll describe what we call marker gene profile analysis, or MGP, with some examples. And I'm going to start fairly slowly. I'm not going to assume a lot of background. I will probably speed up a little bit. So if the background is very basic, bear with me, but I just want to get everyone on the same page. So this is an example of what I mean. So we're starting from very basics here. So where we might, what the context is. So it's very common for people to do transcriptome experiments from where they're trying to look for gene expression differences between two conditions. This is probably 90% of experiments or something like this. And so they have brains or brain samples or brain parts that are coming from one condition, another condition, and they're going to run an RNA-seq or a microarray assay on each one of these samples. And then the output, so the framework for the downstream analysis is this kind of data matrix where we have one column for each sample, one row for each gene, and the numbers in the matrix are an expression level for the gene. And of course, there are 20,000 or so protein coding genes, so this is a very long and skinny matrix. And then for the columns, of course, we have some information about what their groups are, so we know whether they're in condition A or condition B. And of course, you can have much more complicated designs in this, but this is good enough for the sake of discussion. So, of course, what you're trying to find out are things like which genes are differentially expressed between the two conditions. And this is a kind of common paradigm where in genomics and genetics experiments, you're going to get some kind of list of candidates. So you're going to rank your genes. In the case of this kind of analysis, you might be ranking them using something like a t-test, where the genes with the largest absolute value t-statistics are at the top, but you can rank all the genes. And so the challenge is what to do with this. And this is really maybe one of the central challenges in genomics data analysis. It's not specific to neuroscience, and this is something that motivates the interpretation of these lists is something that motivates a lot of the research in my lab. So, you can stare and search. You can, just look at the genes. You can have your student look up each gene one at a time, starting from the top, and decide what to do next. There's methods based on enrichment analysis, often using the gene ontology as a way to try to find patterns, various kinds of what I would broadly characterize as network analysis. And then, so what I'll be talking about is analyses are motivated by thinking about the cell types that are in our sample. And so the key thing to think about for this kind of why cell types might be interesting is that, first of all, there's a few things. So one of them is that cell type proportion changes are a natural consequence of some conditions that we're interested in. And so, for example, in Alzheimer's disease, as neurodegeneration progresses over time, there's obviously a loss of cells, but importantly, this is not something that happens uniformly across the different types of cells. So I took this from this article that is a nice illustration of the idea, where neurons are being lost, And because the total number of, counting of cells within the tissue, so the brain is getting smaller as it loses tissue, but for a given amount of tissue, the number of cells has to add up to 100%. So the relative proportions of other cell types, especially glial cells, are increased. And of course, this is something that's of interest in, even when there isn't something as obvious as neurodegeneration going on, that there might be these kinds of changes. And so I'll talk a bit more about this as well, that cell type proportions are also probably, there's good evidence they vary across samples from different individuals, even if they're normal. And so this is something that we want to try to both address as possibly a confound or as a phenomenon that might be the target of study. So where we're kind of working from here in this kind of study is where you have a piece of tissue that contains many different types of cells. So this is what we call bulk tissue analysis. So of course it won't often be an entire brain, but it might be like say a piece of hippocampus or cortex that contains many cell types. So this is a schematic to try to get some of these points across about what happens. So here I have a toy example where there's two cell types, the triangle ones and the circle ones that make up our sample. So there's only two cell types, and we're going to grind up the sample and extract RNA, and then we do our analysis. And so then we might observe that, well, this gene right here shows a decrease in expression in condition B. But what do we make of this? Because we started out with this complex sample. And there's a lot of different scenarios that can happen, and I'm illustrating three of them on this next slide. So one possibility is what we would call broad regulation. So this gene might be expressed widely, and so here I'm using blue, shades of blue to indicate the expression in the cell types. Maybe it's expressed at higher levels in the triangle cells than the other cells, maybe not, but that in condition B, all the cells are experiencing a downregulation of this gene. So, and so that's often what people are kind of maybe thinking when they do these experiments, is that that's what's happening. Because they ground up the sample and they can't really think, well, how would I know what's going on in any one of these cell types? But another possibility is that the effects are cell type specific. So in this case, in this toy example, that in the triangle neurons, the gene expression is lower, but the circle neurons are not affected. So we would see overall a decrease in expression, but it's actually cell type specific. But it's still regulation. So it's still transcription of the gene is somehow reduced. And so we see less of the RNA in those cell types. But another possibility, which is very important for this talk, is the idea that there might be changes in cellular proportions with absolutely no regulation in the sense that I'm talking about for the other two cases. So in this case, there's for some reason, there's a loss of the triangle cell types, no change in the relative proportion of the circle cell type. And again, we would see some loss of levels of expression of the gene, especially if it's enriched in the triangle neurons. So all three of these scenarios could result in a change in expression in the gene. And again, this is our ultimate output. We don't have direct access to this for these kind of grind up the sample kind of experiments. So the question is, can we work backwards and try to figure out whether one of these scenarios might be a better explanation than another? So this ambiguity is what we want to try to resolve. And as you can imagine, it's hard to do this perfectly because obviously information is lost when you grind up the sample. So it's not an ideal situation. But what can we do? So proportions of cells might be an unwanted source of variation. So if we knew the proportion of cells in each sample, we could correct for it. So we could consider it, for example, as a covariate in a linear model we might be using to try to explain the data. But also group differences in cell proportions might be a phenotype we're interested in. So again, if the Alzheimer's example, if we want to know which types of cells are being lost, can we infer that from the bulk transcriptome. So again, though, we need to know something about the cellular composition. We want to know the cellular composition of the samples that we have. And I guess I'll just pause for a second here to again, further motivate this, that in my lab, we've had a long-standing interest in using, especially post-mortem human brain samples to try to understand things about neuropsychiatric disorders, neurodevelopmental disorders. And it's been a challenge for not just for us, but for the field at large. There's a lot of these studies that have been done. Lots and lots of data sets are available online, but there's really a lot of difficulty in making sense of them. And so we were very interested in whether this kind of approach might help, and we're not alone in that. So just to continue the original train of thought here, that we think maybe we can try to guess something about the makeup of the sample, but why not do this directly? So this is sort of an obvious question, is that for many decades people have used stereology type approaches which is maybe considered a gold standard, where you take sections of tissue, you stain them, or somehow also have some way of identifying the cells of interest, and in a very methodical way count the numbers of cells, and you can come up with density measurements. So that's obviously a lot more work and money. I at least perceive these methods as being very technically demanding. And the other problem is that you don't know which cell types to count if you don't know ahead of time what you're trying to look for. So there are many types of many cell types in the brain, but these kinds of methods, they would typically only focus on one cell type because that's what you can label for at one time. Another problem is that this is a destructive method, so you can't really expect to be doing a transcriptome on this after the fact, so you wouldn't be getting a transcriptome from the same sample. So doing this as well as a bulk transcriptome is something that just isn't done. There are new methods that are emerging for doing multiplexed cell type counting in tissue sections. So these have names like SM-FISH or MERFISH, but these are just coming out in the last couple of years and are not in wide use. And again, there's a lot of bulk tissue data already out there. So bulk tissue analysis bulk tissue transcriptomes is still the way most people do their work. So another technique that people ask about whether it can play a role here is single-cell RNA C. So where the tissue is broken up into individual cells, so there's no in situ anymore, but that you could measure RNA-seq in individual cells if you sample randomly, and then you could infer what type of cell they are based on markers. And then you could just read off the cellular proportions. And again, this is something that I think is probably technically right now not quite the most likely way to go. So the problem is bias in the sampling. If the sampling is biased at all, or not consistently biased, then there'll be problems. But if it did work, if you did get consistent, fair sampling, uniform sampling of each cell type, then this would work. It's just that I think this hasn't been established, and there's not too much known about how the measured, if you did this, how many cells you get, how well that maps onto the original proportions in the sample. One nice paper that came out a couple weeks ago did this in C. elegans, which is a great place to do this because the exact number of cells in the worm and their types is known with great precision. So there's about 1,000 cells in the hermaphrodite. So when in this paper from Jay Schenger's group, where they ground up worms and then single cell sequenced lots of individual cells and then counted up how many they saw and compared them to the proportions that they would expect, they get this kind of plot where, for example, for neurons, where there's something like 300 of them, that they see about half as many as they'd expect based on the known proportions in the sample they started with. So in the paper, they say, well, this is actually really good, that for rare cells, they tend to be rare. And I agree, there's sort of a glass is half full here. But if you wanted to make quantitative measurements, you want to know, how does this vary if I do this for, one worm versus and then another worm and so on. So that has not been established. That's why the multiplex in situ methods might end up being the way to go. But in the meantime, we want to think about these methods that can work from the bulk transcriptomes and work backwards. And so these are broadly referred to as deconvolution methods. And this is based on the idea that, and the previous other types of methods also would rely on this, is that there's some genes that are at least to some degree cell type specific, so they're markers. In that case, the bulk tissue transcriptome contains information on what cells are present because the marker might be a readout for how much of a given cell was present in the original sample. So deconvolution refers to working backwards to infer the input components. And this is not a new idea. We've been trying to trace the literature on this a little bit more thoroughly recently, and so far, the earliest mention of this kind of idea dates all the way back to 1999. This is like the dawn of the microarray era, where, and so this is actually a really famous paper. It's famous partly because the data got reused a lot, but I had forgotten actually that they had this muscle index where, so this is on colon cancer. They had a bunch of tissue samples, and they used, I think, about 10 muscle markers, smooth muscle markers, to come up with a number that says something about how much muscle was in the sample. And I don't think they really used this very much other than to point out that the samples with N here, the normal samples, tended to have a lot more muscle in them. So the numbers are higher. And so moving to brain applications, and again, trying to trace this back and give you guys an idea of how well established these approaches are, is a paper from my friend and collaborator Etienne Sibyl and others, where they, again, I think this, I don't know if there's anything earlier than this, so I'll tentatively give them credit for this concept of trying to compute a white matter, gray matter ratio from bulk tissue samples. So what they did is they took white matter from human brains that they were also measuring cortical gray matter expression profiles for, and looked at how similar the white matter expression patterns were to their the gray matter samples, so they could get this sort of ratio. So this is estimating the presence of transcripts that look like they come from white matter and gray matter samples, and they attribute some of this to possibly the contamination, that when you dissect, maybe you get a little bit of white matter by accident. But there's also, they presented evidence that this is an inherent variability across individuals. So if you look at samples from the same person for two different brain regions, that this white matter ratio tends to be concordant. And so these exceptions here might be ones where there was just contamination. So, so this is obviously quite crude in the sense that it isn't cell type specific, but obviously white matter is heavily enriched in glial cells compared to gray matter. So this is something about variability in glial content across samples. Another paper from around the same time period that at least approaches these ideas with just some observations, interesting observations, comes from Oldham et al., where this was a paper looking at co-expression patterns in normal human brain in cortex. And what they observed is that when they clustered gene expression profiles, so this is showing expression profile across samples, so expression levels, and they used a computational method to identify clusters, they noticed that a lot of the clusters were enriched in what we think of as cell type markers. And so the variability in the expression level of these kinds of genes from sample to sample and their consistencies, you have a whole bunch of genes that are associated with the cell type that are going up and down across the samples. They interpreted that this is happening because of differences in the cellular makeup of each sample. And so, and I point out here that, obviously stereology studies or the direct counting, this is inherent also, is that there are individual differences. So this is one of the papers that, together with Etienne's paper, really made me think that these kind of approaches, that there really are signals here that reflect cell types, and that the variability we see in expression within samples of that are normal or disease, that even that variability is due to these compositional differences, and of course that group differences might be, you can pick them up in the transcriptomes. Okay, so how do we put these ideas to work? And I hope people are following me, and this is part of the mystery of webinars, is that you could all be checking your email and doing other things I had no idea. I forgive you. So I'll forge ahead. So if we want to try to put these ideas to work and try to do deconvolution, so we don't need a whole lot of information, although there's lots of gotchas and cautions here that we can talk about. So first of all, we need some kind of cell-specific markers or a complete expression profile for a cell type that we're interested in. And of course, then we need measurements of those markers or profiles in the bulk tissue, because that's what we want to study. And the output is some kind of estimated proportion, either in relative or maybe even absolute terms, of the cell types in the samples. And as an aside, and I'm not going to talk about this very much, is an alternative approach that also you'll see people do is where they don't try to do proportion estimation, but they'll use cell type specific markers in an enrichment analysis. So instead of using the gene ontology, or in addition to using the gene ontology, they might look at whether genes that are associated with a particular cell type are enriched in their hit list. So this is obviously a bit more indirect than trying to estimate proportions, but it's a similar idea. So, and again, to sort of highlight that this is this is not a new idea. It's an active area of research to develop methods for doing this kind of deconvolution. So here's a list of some that Leela put together, based on some of the literature. And I'm not going to go through this. It's just really to point out a couple of things. So one is, first, is a bunch of methods. They use a variety of statistical approaches, but they're all basically trying to come up with a model based on marker genes or reference profiles that can be used to explain the bulk tissue data in turn. So where the parameters are going to be something about weight, like weights for each cell type. And so those weights are somehow related to the proportions. And so another thing to point out here is that, so while these methods and some of their application methods are well established, most of the applications tend to be in tissues like blood, where there's a liquid tissue, that it contains different cell types, and there's lots of existing methods for measuring cell type proportions. So there's lots of isolating cell types, lots of markers already for blood cell types. So you'll see a lot of papers that use, especially these kind of methods development papers, will often be using blood. And that these methods, or ones like them, and the ones I'll talk about more in a minute, is that this kind of assessment or correction for cellular composition is really routine, especially for in DNA methylation studies, where you can do the same thing with methylation patterns. That changes in methylation, and one reason this is important is methylation changes tend to be smaller than transcription changes. So so small offsets, in cellular composition will really have a big effect. And also you see it used in things like expression QTL studies, where it's considered a nuisance variable source of variance, that there's differences in cell type proportions. So if you don't know much about these kinds of data, that's okay. It's just an aside. So in brain, there are, this is just a sampling of some of the papers that we've found where people have used methods like this in brain. And again, there's a variety of approaches and angles that people are coming from when they do this. So we see ones in Parkinson's and Huntington's disease, neurogenetic disorders, Huntington's disease, in aging. I'm sure there are others. So I'm saying all this to really kind of make sure it's clear that this is not like some crazy idea that we dreamed up, that lots of people are doing this. So just to summarize so far, variance in cellular composition exists across individuals and conditions, and it's an established concept that cell type specific transcript levels in bulk tissue give a readout of how many of those cells are present, at least relatively. And there could be discussion about how true this is, and I think it's an interesting discussion to have. But we can be a little careful about our interpretation, but this should improve our interpretation of the data. If these two statements are true, that we can get some readout and that this variance exists, then this has got to help us understand the data. So what we need, though, are markers, lots of markers for lots of cell types. And if you look in the literature, of course, lots of neuroscientists could probably name some markers for their favorite cell type. So there's standard ones that have been around for many years, but we want more of them. And so where do we get those? So the way we do that is using cell type resolved transcriptomes. And so this just refers to any kind of method where you have an identified cell type that has been isolated, and then the transcriptome is measured so that you can compare that to other cell types and see what's specific about the cell type. So identification, there's many methods for doing this. So transgenic markers, immunohistochemistry, using one known marker to try to pull out the cells. Track tracing is another method that's been used. And then isolation, so laser capture microdissection or fluorescence activated cell sorting, manual sorting. These are all methods that people have used to get the cells out. And then, of course, we measure transcriptome using either pooled cells or individual cells more recently. And we're taking advantage here of the fact that many of these data sets are publicly available. And so we're very grateful to the people who've collected these data sets, often with a lot of labor and cost. So most of these are in mouse. So of course, you want to apply this to human as well. And so we're going to use, so there's, obviously that has some risks, taking mouse genes and using orthology relationships to look at them in human, but we can do some checking to make sure that things are working okay. But yeah, this is definitely one thing that we would like to see more human data. So what we've done in my lab is that we've gathered up these data sets, as many as we can find. We've cleaned them and quality controlled them and then harmonized them. And there's two types of data sets that we've been using. Most of the data sets are microarrays where they're using purified cell types. So they're pooled cells, not individual cells. And then we've used some single-cell RNA-seq data. And the problem with using a lot of single-cell RNA-seq data sets, if you're familiar with those, is that they often don't, mapping the cell types they identify to quote-unquote known cell types is sometimes challenging. So we have to be a little bit careful there because they're often being used to discover cell types. And so we've been a little bit cautious in how we approach this, and we want to use cell types that at least other people believe are real cell types. So of course this can be amended as we go forward. So this little schematic just gives a little bit more information. I'm skipping a lot of details in this presentation because I just want to show you, give you a high-level overview. But we're taking this data that, in this case, this is a single-cell data set that we're using comes from the Allen Institute, and where they clustered the cells and then identified what they thought the cell type was for at least some of the clusters. So again, mapping these to known cell types. And likewise, there's publicly available microarray data for purified cell types. These are generally already identified cell types. So we had to harmonize these to get all these papers together in terms of cell types they're talking about, as well as the RNA-seq. We've also tried to do this in a brain region specific way. So we don't want to assume that astrocytes, for example, from cortex are the same as astrocytes in hippocampus. In some cases, we've sort of had to do that if we want to use that cell type in multiple regions. But where we can, we try to use brain region specific data. And of course, some cell types are only present in one brain region. Another reason this is important is that it's unlikely, if you want to try to find a marker that's specific for a cell type, it's very difficult to do that if you require that it not be expressed in any cell type anywhere in the brain. And if you constrain it to be anywhere in the body, it's even worse. So for example, there are genes that are expressed in one cell type in the cerebellum that are good markers for them. But those cells, the same gene is also expressed in certain cells in other brain regions. So it's a good marker for cerebellum, one cerebellum cell type, but it wouldn't be a good marker if we were trying to combine it with another brain region that expresses that marker in another cell type. So we're assuming that most people are doing something relatively region specific and not taking entire brains and putting them in a blender. But you can work with that too if that's all you have. It just gets harder to find specific markers. So then we select markers, and I'll talk about a little bit more about that. And also the data is what's made available through a web interface. So let me just show you the web interface real quickly. So it's not very exciting. It's really fairly straightforward presentation of the assembled data. So again, we've aggregated data across many different studies and put it here so you can search for it. So you can search for a gene. You can constrain this to be one region. So here I've selected cortex. And each point here reflects the measurement of this gene in one cell type in one study. So and the circles are for microarray studies, the pluses are for RNA-seq. And so you can see the degree to which the gene varies across the studies, whether the gene looks like it's relatively specific or not, or whether it's expressed in a given cell type or not. So you can, if you hover over these points, this is not a live demo, you can see the citation for which point this is coming from. So we think this is quite valuable on its own, but really the underlying data are where we see a lot of the value, and applying it to analysis, which is what I'm going to talk about next. And so there's links to the other resources, so especially the GitHub repository where we have the marker gene lists and some of the R code that we've developed to put this to work. And so here's just a list of all the cell types that we have so far, and here it's broken down by brain regions. Like I mentioned, we've tried to make it brain region specific where we can. So for some cell types, we just sort of assume they're equivalent all over the brain, which is, of course, questionable, but that's what we can do. I think there's close to 40 cell types on this list. We're of course interested in adding more data, and it's really great when we have multiple data sets for the same cell type, because of course they don't always perfectly agree with each other. And so in this kind of meta-analysis, we want to get the best information. Okay, and so then for selecting markers, again, I'm not going to go into details of how exactly we do that, and you can look at the details of that in the preprint or seen in the paper. But basically, we're looking for genes that are, as much as possible, within a brain region, expressed at reasonably high levels in the cell type of interest and not in the other cells. And so if we make a heat map of the expression levels, these are relative expression levels for some of these markers, you just get an idea of what we end up with. So there's more markers here for some of the cell types. We just cut it off to fit on the screen. So here's a bunch of genes that are expressed at high levels in astrocytes, and they're basically not expressed or much lower in other cell type samples, and likewise for other cell types. And of course, as you can imagine, as you can see, we have different numbers of samples. So that for here we only had 3 samples to use for in this case microglia, and the number of markers we actually can find and their quality does vary from cell type to cell type. So that's sort of a general something to be aware of, that when you use this, of course, the quality of the markers is going to have an effect on the outcome of the analysis. And so one of the important features of this is that we're not advocating the use of single markers, but that you look at lots of markers at once, and that coherent patterns of markers is much more convincing than the behavior of a single marker, both because the markers aren't perfect, but also because the data that we're working with is often pretty noisy. And when you have a cell type that's only a small proportion of the tissue in question, of course, it's going to be even noisier. Okay, so we have attempted to at least, so because some of the markers that we're proposing some novel markers, it's sort of important to validate these, and we tried to do this as much as we can. So of course, some of them are previously recognized as markers. They're not controversial. Interestingly enough, some markers that people have previously reported don't really hold up that well in this data. So then others we've able to use in situ hybridization, either from the Allen Brain Atlas or through work by Etienne Sabille's group, where we can confirm that the specificity holds. But of course, we haven't independently confirmed a lot of the markers. And so we do expect that there'll be adjustments as new data come in. And, but in all cases, we're using multiple data sets, and so we're pretty confident that most of these are going to be are pretty good. Okay. So how do we put this to work in the data analysis? And so this is where this concept of marker gene profiles, or MGP, comes in. This is the sort of deconvolution-like aspect, although we don't call it deconvolution, and I can explain why. But basically, an MGP is a summary of the expression levels of marker genes for one cell type in a particular data set. And then we're proposing that we can use this with some caution as a proxy for relative cellular proportions. So the way this works is that you start with, so for example, for dopaminergic neuron markers, So again, NeuroExpresso is based on mouse. So we have some markers for dopaminergic neurons that we can map by orthology to human. And so we might lose some genes there if they don't have orthologs. And then we look at the expression levels of those the genes in the data set that we're interested in analyzing. So this is just a plot for one data set of how some of these markers behave. And so we can see some of them are expressed, for whatever reason, are expressed at very low levels or show patterns very different from the others. And so we would prune out some of the markers, or at least mathematically reduce their impact. So we're looking for a consensus here. So we actually use principal component analysis of this pattern to identify sort of an eigengene reflecting a summary of the expression of the markers across the samples. If you don't know what PCA is, that's okay. You can just think of, like, if we just average these, we get some kind of pattern. And we end up with a number for each sample. So if we just plot that, and I'm not, it might be that these data, this is sort of a schematic, so these data might not match up, so don't look at it too hard. But that we have individual measurements of MGP for, this is for one cell type, for each sample. And of course, we can consider whether there are group differences between at the MGP level. So in this case, say these samples were from Parkinson's patients and these were from normal, and that we see the dopaminergic marker gene profile is lower in Parkinson, which is what, of course, we might expect given the degeneration of dopaminergic neurons. And of course, you can generate this for a bunch of different cell types at once and look at them. So these can also, I mentioned, can be used as covariates in an analysis if you want to sort of correct for this and look for changes that might be happening in addition to changes in cellular proportion changes. So I mentioned, like, there's a lot of people have applied these methods, but we still felt it was important to validate this as much as we can. It's the validations I'm going to show you are fairly straightforward, I think. So in this case, we've looked at gray matter versus white matter, and all of this is based on published data sets. So we're just taking those data sets and reanalyzing them. And so, of course, in white matter, there's fewer neurons and more glia relatively, and so that's what we see. So for all the neuron types that we test, we see lower levels in white matter than in cortex, and opposite for glial cell types and other cell types. So that's good, but not terribly exciting, or it's fairly obvious. So a little more interesting is where we've been able to find some cases where we have cell type counts based on stereology for at least one cell type, and that we can use to cross-check for the same samples where we have transcriptome data available. So this is from a paper by Uranova et al., where they counted the number of oligodendrocytes in, I can't remember which region of cortex they looked at, but we have transcription profiles and therefore MGP estimates for oligodendrocytes for the same region, or a very close region. So obviously not the same sample because these are both destructive methods. And the group differences that they reported, this is just redrawing the data from their paper, where they say that there were lower levels of oligodendrocytes, lower counts in these conditions. We'll recapitulate that with the marker gene profiles. Again, the same data set. So unfortunately, we don't have counts for other cell types from the same data set, and so this kind of data is rarely available. But where we've been able to look at this, it's held up quite well. Okay, so then the other thing that we present in the paper, and which is sort of a validation, but also I think interesting in its own right, is applying this to a neurodegenerative disease, Parkinson's disease. So just whether MGPs capture is already sort of indicated in my little schematic that they do. But what's less clear is, so we know that's happening, is the degree of transcript regulation. So if you take someone who's died, they take their brain, and they died with Parkinson's disease, is there something else going on in the substantia nigra besides loss of neurons? And so previous studies, where we've been able to get data for this question have suggested that there is regulation going on, but they were not taking account of the fact that cell type proportions are changes. So which of these things is going on? And so we've reevaluated that. And so first of all, this is the validation part, where this is from 3 different data sets. One of them had 2 sort of sub data sets. We see this nice reduction in our estimate of proportions of dopaminergic neurons in all of the data sets. And this is a good time to point out that these numbers on the y-axis here, you mustn't read them off as proportions. This is not saying that, that 80%, this is not 80% or something like that. This is a relative, it's actually scaled between 0 and 1, so it's relative. The amount of change this corresponds to is something that we can't really get at very easily on a case-to-case basis. We could try to make some estimates, but it's best just to think of it as a relative proportion. So you can't use it in an absolute sense. Okay, so then, so is there regulation as well? So one of the papers where they had proposed a signature that they referred to as reflecting regulation in Parkinson's disease. And so we looked at these genes and we find that they're actually all, or almost all of them, are highly correlated with the dopaminergic marker gene profile. So that's what's shown here for the principal component of their signature genes. So this suggests that they're actually like weakly markers of dopaminergic neurons. So they might not have been good enough to actually make it to our marker list, but they're highly correlated with other markers. And this is true for both the controls and the individuals with Parkinson's disease. So I think this would be the Parkinson's disease one, the green ones. So that suggests that these are really just genes that are expressed, enriched in dopaminergic neurons, and so their expression levels are tracking the numbers of dopaminergic neurons. So we think that, to a large degree, we explain this signature as simply a change in proportions, and that there isn't any really good evidence for interesting regulation going on. So that's not definitive, but that's based on this analysis. So the last thing I'll do is just give you a very quick high-level overview of how you can do this yourself if you're interested. If you have a data set where you want to apply the analysis, so you can use the markers that we provide, or you can make your own list if you don't like our markers. So we provide an R package and the supporting data, so the gene list, and the key method in the R package is this MGP estimate method. And it takes as input your expression data matrix, the marker gene lists, and then you can have a sample grouping factor as well. So for example, Parkinson's and control. And the output is this estimations object, so which includes the estimates for the MGP values for all the cell types that you included in your analysis. And then of course you can use that for downstream analysis. And it, because there's various things that can go wrong with markers not being expressed or whatnot. So there's some quality control and sanity checks that we provide to make sure that markers are behaving well. Because unlike some analyses that people apply to data sets like this, we really think this has a fairly direct interpretation in terms of cell type specific effects, either cell type specific expression changes or more likely proportion changes that, there's some expectations for how things should be behaving. And so you want to make that direct interpretation, then you can be cautious and look at the data quite closely. So, okay, so in terms of cautions and caveats, so I mentioned that NGP is only a relative measure. If you're going to use the eigengene approach, you really need enough samples. So you could use some other way of building an MGP rather than principal component analysis, or use the enrichment type approach, which isn't so good for comparisons between cell types. So you also need enough markers. So really, if your cell type doesn't have enough markers, or they weren't measured in your data set for some reason, that's a problem. But we really like, minimum 3 is the bare minimum, but ideally you'd have a lot more. And of course, that there's not much sense in trying to measure cell types that you know are not present. So, including a Purkinje cell profile, if you're measuring amygdala, it wouldn't make a lot of sense. So just to sort of wrap up, we are actively expanding this, so both in terms of adding more data and more software, but we're also applying this. So we have what I think is a really interesting story in cellular changes in schizophrenia and bipolar disorder, which if you're going to be at SFN, there's a poster that Leela's going to be presenting. We also used the cellular markers, the NeuroExpresso data set, for comparison to electrophysiological features, and that's being presented at another poster, 750.11, as well as a paper that's under review. And so just to summarize, I've hopefully convinced you that cell type makeup variability is a source of variance in brain genomics, that taking that into account gives us an additional level of insight into complex data sets that we think is really valuable. NeuroExpresso, our contribution there is the data and tools that help put this into practice. So you could, the marker genes we provide, you could try to use some other people's tools. We're not forcing one way of doing this. There will be a poster specifically about NeuroExpresso at the conference next month. If you're taking the short course, you'll get this again in more detail. And please get in touch with myself or Ogun if you want help with using the data. And I'll just end by thanking especially Leela, Ogun, and Srijoy, who really did most of the work, and other members of my lab who participated one degree or another, and our funding, especially from the NIMH, and I'll take questions.
Hi, Dr. Pavlidis. This is Devin again.
Hi.
Thanks for a great webinar. We do have a few questions. I can start with Dr. Pollock. How are you coordinating this effort with the brain cell census effort?
So there's obviously a lot of projects that are collecting more data for different cell types, and we're not directly coordinating with anybody, although, we're looking for data sets that have been made publicly available. So as they emerge, we are trying to incorporate them. If there are data sets that you think we should be using that already are out there that we just missed for some reason, it'd be great to hear about them.
Great. And moving on up the list, Dr. Bandrowski, I think, asked a question a little bit earlier in the chat. If you can find it, it's a bit long, or I could read it out to you.
Oh, okay. I closed the chat because...
Oh, no problem. I can read it. I can read it. She says, "Hi, Paul. Asking for comment on this topic. It seems like with cell type specific regulation information is giving us a lot of additional information. But mapping onto classical cell types is a confirmation or refutation of classical anatomy cell types. How much more have we learned with genetic approaches compared with classical anatomy? And is it worth it?" Yeah.
So if I can just sort of rephrase that, hopefully I get this approximately right. This is about how do we define cell types in the first place, and whether the cell types that are classically recognized are really cell types. And also there's a lot of ongoing work using especially single cell approaches that are claiming to identify new cell types. So I think obviously the people who are collecting the data, I think the answer is they both have some validity, but it really comes down to how you define a type. And so there really doesn't make sense to define a type purely on genomic grounds without having some kind of correspondence to either morphology or function or connectivity, something that I think most neuroscientists would probably agree that's got to have there's got to be something else going on rather than just finding some, one gene, some other explanation. It's not satisfying having a cell type that's purely defined based on a transcriptome. It might raise a hypothesis if you've identified a cell type, a possible cell type difference, but it won't be accepted as a canonical cell type until more is known about it. And that's one reason that we've been cautious about incorporating just clusters that people have identified in single cell sequencing into NeuroExpresso, because we don't know what they mean. Matching them up across studies is a little bit problematic. So we're just sort of waiting. Maybe that's a mistake and we should just go ahead and say, here's clusters 1 through 100 from the single cell sequencing study, this or that study, and just add that in. But, we're worried that's going to be a bit of a mess. So yeah, I think it's a discussion that a lot of people are having right now is how to define cell types based on these new kinds of data and what they mean. So it's a very important question.
And I also received a question in the private chat. We had a participant wondering if you could elaborate on the course that you'll be teaching at the Society for Neuroscience.
Okay, I'll try. I can actually the abstract for that. So this is organized by Jane Roskams in collaboration with SFN staff. And so it's, as you can see, it's taught by a bunch of different people covering many different areas of neuroscience. So there's Alan Evans, who does brain imaging, so he's going to talk about, I think, distributed computing. We have Terry from Trygve is going to be talking about single-cell RNA seq, I think. So there's a bunch of different topics. So they're all going to be given like a little slice of time. And then in the afternoon, there'll be a set of hands-on exercises. And there'll be, I think, time for people to pick 2 of them that they want to do. So in those breakouts. And so we'll be, there's also what they call a syllabus that's going to be provided for that as well. That contains sort of like little articles that explain the material, and that will be made publicly available after the course. And so that actually has almost like what now is, for the moment, the best user manual for NeuroExpresso for the R tools. But we can share that with people if they just ask us ahead of time.
Great, and I think we have 2 more questions. This one's from Koei. You may have just touched on it, actually. She says, you mentioned about the short course. Is that available online or at the Society for Neuroscience this year?
Well, so yeah, if you go to it's on-site only, and it's on the Friday, right before the main conference starts. I think I don't know how much the registration costs, and it depends probably if you're a student or not. But it's not very expensive. So the hotel, extra night stay in hotel will be more. And so yeah, I assume I don't know how many slots are still available. You'll just have to see if you can still register for it if you're going to be able to make it. But yeah, I definitely encourage people to check that out.
And Yuling says, thank you for the presentation, very informative. She's wondering if you could talk a little bit about how to standardize, validate, combine the data pulling from various data sources and papers generated by different teams.
Yeah, it's a challenge. And, like one of the things that it's definitely a problem. People use different platforms. RNA-seq is quite different from microarrays. And so, again, the details of how we do that is provided in the paper, the preprint. But it's basically a process of trying to find a common scale for things as much as possible. So, and where we can't do that, we keep the data sets separately. So actually for RNA-seq, because it's scaled so differently than microarray data, that we kept it separate, and then we select markers for that separately from the microarray data, and then kind of try to reconcile the markers across them. So that's one thing. For the different microarray data sets, it was something that we're kind of more in general comfortable doing, or for multiple RNA-seq data sets, it'd be more or less the same. So one is aligning the cell types. So figuring out like what cell type are they talking about if they didn't define them exactly the same way. Mapping genes across different microarray platforms, that wasn't so much of an issue because most of the studies, I think, use similar platforms. Cleaning the data, so removing things that look like obvious contamination and other kinds of artifacts. So yeah, I mean, that was actually a fairly big job that Ogun undertook, and yeah, I think giving a full accounting of that would be difficult right now. But there's information. I mean, in general, it's something that I have a fair amount to say about that we do routinely, and I'd be happy to take a discussion offline. If you're trying to do that yourself, we could certainly advise you.
Great. Well, I think that's all for our Q&A. I just want to say thank you once again to Dr. Pavlidis for the informative webinar. And to all of our participants, thank you for joining. We will be here same time next week with Dr. Greeth talking about NIF and DKNet making research tools FAIR capital fair. Great. Thanks again.
Thank you for having me.
Yeah, absolutely.